Good morning, everybody. I am happy to welcome everybody to the seminar series this morning, and it is my pleasure to introduce Dr. Martina Bazzaro, who is giving our talk today. Martina received her PhD in Italy in the Department of Pharmaceutical Sciences and received her postdoc training at Johns Hopkins in the Department of Pathology, and then joined our faculty here in the Department of Obstetrics, Gynecology, and Women's Health, where she is now a tenured associate professor. Martina's research focuses primarily on ovarian cancer, although I think we'll hear some other areas, maybe newer areas for her today here. She's been highly productive and very well funded in this area. She does really beautiful work, as you'll see today, dissecting sort of mechanisms of ovarian cancer development and progression, with really the goal of enhancing treatments for ovarian cancer patients that have chemo-resistant disease, which is really a huge challenge in the ovarian cancer field, so highly significant work. One of the areas that she works on that we'll hear about today includes understanding mechanisms that regulate microtubule dynamics via a protein called UNC45A. So thank you so much, Martina, and it's all yours.
Thank you for the very kind introduction, Kelly, and for the invite, and for being here. I think we need a little bit of less light. If we can turn the lights on a little bit, off a little bit. So today I'm going to talk about one of the research projects that we have ongoing in the laboratory that has to do with the role of a microtubule severing protein UNC45 and the role that this protein has both in cancer, and we would use ovarian cancer as a model. That's a model we have used, and neurodegenerative diseases, and we will talk about Alzheimer's disease in particular. The very first thing that I want to do is I want to acknowledge the people that actually have done the work, as well as the collaborators and funding agency that will come in the next slide. So most of the work that you are going to see on the cancer aspect of UNC45 was done by Ashley Mooneyham. Ashley is a former grad student in the MICA program, and by Yuri Habik. Yuri is a former postdoctoral fellow from the University of Tübingen in Germany. And then most of the work that you will hear about on UNC45 and how it regulates neuronal microtubule stability was done by Asumi Oshino. Asumi is a former undergrad in my laboratory and now a grad student at the pharmacology graduate program here at the U, and by Valentino Clemente, a postdoctoral fellow from the University of Bologna in Italy. Collectively, they have published a number of papers that we'll kind of go through today. As far as funding agency, the DoD, the NCI, the Alzheimer Association, and the Minnesota Ovarian Cancer Alliance, and as far as the collaborators that have helped us to learn how to deal with microtubules and how to ask questions about microtubule, David Oddy and Melissa Gardner, and then Juan Troncoso at the Johns Hopkins Institute that has provided us with clinical samples of Alzheimer's disease that we could then use to see whether or not UNC45 has a role into that in developing of AT. So while cancer and neurodegenerative diseases have been considered at the opposite end of the spectrum of human diseases, because they have very different characteristics. Cancer cells, they keep dividing and they resist death, while neurodegenerative diseases are associated with the death of post-mitotic cells, right? Neurons are post-mitotic cells. Today, genetic and cell biology studies show that there are multiple common pathways and proteins that are common that regulate both cancer and neurodegeneration. And that include microtubule associated protein that regulate microtubule stability. So the most, I don't know why I can't. Okay, so the most famous, the most common example of microtubule-associated protein that is involved in both Alzheimer's disease, in this particular case, and cancer is tau. So if you look to the left, tau is involved in what a group of neurodegenerative diseases that they take the name of tauopathies. And basically what it is that you have microtubules inside the cells. So microtubules are in this picture, these green, hollow pipes inside the cells. Tau is this blue string. So tau binds to, under normal condition, let's put it this way, tau binds to microtubules and provides them with the required stability for microtubule to do what they have to do inside neurons. However, there are conditions under which tau get hyperphosphorylated, and when tau gets hyperphosphorylated, it comes off the microtubules and it forms filament and tangles. But what matters is that as it comes off from microtubules, those microtubules are not protected by the microtubule stabilizing effect of tau any longer, and so they essentially fall apart. And so you have loss of microtubule mass, which is one of the characteristics of tauopathies. And that's what tau typically does in neurodegenerative diseases. Tau in cancer also plays a role, because in particular in taxol resistance, because tau and taxol, they bind to the same site of microtubules. Okay, so if you have microtubules that have a lot of tau in it, then when you treat the cancer cells, for example, with taxol will not be able to bind efficiently to the microtubules, and so it will not be able to do what it needs to do in order to kill the cells. In fact, tau expression is a biomarker for taxol sensitivity. So if you have a clinical specimen, if you have a cancer that has a lot of tau, that cancer is less likely to respond to chemotherapy, to paclitaxel as a chemotherapy. So that is just, now today we're not going to talk about tau, but it's an example of how this class of proteins, the microtubule associated protein that regulate microtubule stability, are known to play a role in both cancer and neurodegenerative diseases. So today, my objectives are to talk about microtubules a little bit, to give you a sense, you probably know them, but to give you a sense of what they look like and what they do, how we have discovered that ANC45 regulate microtubule stability, the data that we have in terms of how ANC45 plays a role in cancer and in neurodegenerative diseases, and then we'll briefly talk about therapeutic opportunities that targeting ANC45. So microtubules, that's what they look like. So microtubules are the stiffest of the biological polymers. They are as stiff as plexiglass. They are formed by alpha and beta tubulin subunit. So alpha beta tubulin subunits, they organize, they assemble longitudinally to form the protofilament. And then this protofilament, they assemble laterally to form the microtubule wall. Typically, under the right condition, 13 protofilament will form the wall of a microtubule. In terms of size, microtubules are actually quite big structure if you think about it. So their diameter, internal diameter is 19 nanometer, the external is 23 or 25, but they're extremely long. Actually, they can be up to 50 micrometers, which is really, really long if you think about a cell, right? So just to give you a sense of proportion, the thickness of a human hair is anywhere between 30 and 50 micrometers. And then you have cells that also really vary in size. For example, cancer cells will be anywhere between 30 and 70 micrometers. And so if you think about a microtubule being 50 micrometers, it's actually quite a long structure. And then, of course, you have neurons. And in neurons also, they vary in size. You can have very, very small neurons, 5 micrometers is typically the size of a cerebellar neuron, but then you can have huge neurons, like neurons that can be 1 meter long. So motor neurons, which are the ones that are born in your spine, and they go all the way down to your leg to make your leg move, can be up to 1 meter if you have very long leg, that is. Microtubules have a polarity, right? So they don't look the same at their end. So they have a minus end, which is the place they're born, And so they are born at the mitotic organizing center, that is the minus end, and then they grow, from there they grow to give rise to the other end, which is called the plus end, which is the place where they grow. Microtubules are dynamic structure, so don't think of microtubules as something that is built and it stays that way. They constantly grow and shrink from the plus end, undergoing what is called dynamic instability. And so what you're going to look at this here, when I start this video, so those are cells where the microtubules have been labeled with the green protein that is called EB1. EB1 is a plus end tracking protein, which means that basically it decorates the microtubules, but it accumulates at the end. And so you will see how this green end, this green tips, they grow and they shrink. They go and they shrink. That is the dynamic instability that is the characteristic of the plus end of the microtubules. Now, microtubules are also dynamic and unstable for other reasons, including the presence of microtubule severing protein. So what microtubule severing protein do, they cut the microtubule along their lattice. So instead of messing up with the growing and shrinking of the plus end, they cut microtubules like scissors along their length, generating again minus end and plus end. So this is the plus end because they grow. This is the minus end. When they get cut, you still generate a minus end and a plus end. Microtubules, obviously, sorry, I have to get my glasses. I can't read the screen. I see. So microtubules, obviously, they play a number of different roles in both dividing cells and post-mitotic cells. They are obviously important for cell division, and what you're looking at to the left, top left, is mitotic cells in metaphase. So there, microtubules, they form the mitotic spindles, which are necessary for the proper segregation of the chromatid, the sister chromatids. And you also have other kind of microtubule that allow the cells, the mitotic cells, to be in the correct position for the separation to occur. Then you have microtubules being very important in cells when they are actually moving, so in interphase. They are able to actually reach out within the cells into the area where the cells they have to move, and then they retract when the cells stop moving. So they are also important for structure. So, right, when we talked about how neurons can be cells that are up to a meter long, so you can imagine the kind of structure that these cells have to maintain to be able to do what they have to do and to be able to go, those axons, for this axon to be able to go where they have to go. So that is what is keeping the structure, what is considered the skeleton of neurons are actually microtubules. They're also important for transport, in particular in neurons, because everything that goes from the soma to the periphery and comes back goes through the microtubules. So ANC45, it's a protein that is very conserved throughout evolution. It was first identified in worms and flies, and the reason why it's called ANC45 is because it comes from a mutagenesis screen where the dish number 45 had a mutation in this protein, and the worms couldn't move properly. So they moved in a funny way, in an uncoordinated way. That's why the name comes, ANC45A. Or sorry, ANC45, I should say, in the flies and in worms. They only have one type. Vertebrates, we have two forms. They are the product of two different genes, ANC45B, which is involved with muscle and myosin regulation, and that's not what we're going to talk about today, and ANC45A, which is expressed in all cells. In terms of what it looks like, it's a 944 amino acid protein, 100 and some kilodalton. It has an N-terminal domain, which we have recently shown is required for binding to microtubules. It has a central domain that is considered to be required for band conformation. This is a protein that bends and oligomerizes, or at least the worm isoform of it. And then it has a C-terminal domain that is required for myosin II binding. So now Ashley, our grad student in the MyCap class, did a lot of work to show that ANC45 is a microtubule-associated protein. We did that by applying turf microscopy. So basically what you do, you go to the slaughterhouse, you take brains from pigs, and through a very smelly process in the laboratory, really, you end up with microtubules from brain, porcine microtubules. Then you label them with rhodamine, they become red. And then you basically put them on the top of a glass that has been coated with anti-rhodamine so that when you put the red microtubule on it, they stick. And then you put your protein of interest, in our case, ANC45 GFP, on the top of it. And then you do turf, which is a fancy microscopy where the light comes with a very shallow angle, so basically you don't get a lot of background. You only see what is very, very close to the surface of the glass. And so this is what we saw. So when you put ANC45 on the top of microtubule, ANC45 decorates the microtubule with what we are now looking at, a very interesting periodicity, with a certain periodicity. It seems like it binds to discrete areas in the microtubules, but definitely it can be considered a microtubule-associated protein. What does ANC45 do to microtubules once it binds them? It breaks them. So to the left, you are seeing those microtubules, and in this picture, you're going to see microtubules only. There is no green channel on it, so you will not see ANC45, but this is done either in absence or in presence of ANC45. And I believe here is 250 nanomolar. So when you add ANC45 after a certain period of time, and we're talking about 5 to 10 minutes, that's when the process starts, see, this is what happens. So you start to see not only that the microtubules are shorter, so you have a loss of microtubule mass, but they also seem to bend. And in fact, if you look at the video over time, you will see that in presence of ANC45, the microtubule, they bend and they break. The same can be seen here. So in here, you see both channels. So you see the microtubules in red, you see ANC45 in green, and you can see now, this is a very small area we're looking at. So you are not capturing the bending, but you are capturing the breaking in presence of ANC45. So ANC45 binds to the microtubule, the microtubule break, and then eventually ANC45 seems to remain attached to at least one of the microtubule's ends. And that's why I told you before the microtubules have a polarity, minus and plus. We don't know to which end ANC45 binds or remains bound to once it severs microtubule, but knowing that would be important because the different ends have different roles, of course. So this is what happens in vitro, right? So this is what happens to microtubules in presence of ANC45, to porcine, to what we call it ex vivo or in vitro microtubule in presence of ANC45. Now, what happens to, okay, so this is the video. So you're going to see, when I start this video, so those are microtubules. This is probably a video at minutes, maybe 2 or 3. So there is already some ANC45 in the chamber, you see it. As the video starts, you will see what happens over time to those microtubules. It's going to be difficult to see all of them. I would suggest you focus on this one because it's right there and you will see what happens to it. ANC45 binds and the microtubules get shattered. And you end up with like 30 minutes, 0 microtubules left. Now the question is what happens in vivo? Oops. Okay, so first of all, the first thing that we need to look at is, okay, so now ANC45 is a microtubule associated in vitro is it a microtubule-associated protein in cells, right? So we looked at both interphase and mitotic cells, and this is endogenous levels that we're looking at. So you can see ANC45 is present on the microtubules in red here, and interestingly, it has a dotted, almost dotted pattern, which is very similar to the dotted pattern we have seen in the purified microtubules. And then, of course, it decorates the both interphase and microtubule in the mitotic spindles. And we could even capture actually ANC45 to be present at the mitotic spindles in a dividing cell in a clinical specimen in situ. Question is, what happens to these cells when you overexpress ANC45? So those are RFL6 cells, those are fibroblasts. We use those because they have a very flat morphology and because their microtubules are well distributed. So you can actually see single microtubules, behavior of single microtubules. It's very difficult to see that in any other cell line, to be honest. So these are GFP, so you're looking at alpha tubulin, right, staining, but these are GFP infected cells and these are GFP ANC45 infected cells. And so those are fixed cells. So if you look over time, and this is 24-hour post-ANC45 infection, you see a dramatic loss of a microtubule mass in the ANC45 overexpressing cells as compared to the controls. This is a still image of a part of a movie, and you are basically looking at the same phenomena, with the exception that you can see the bending, because we are now capturing one single microtubule, and you can see the bending, and you can see the breaking of that microtubule following bending. And you can see that even better if I showed you a movie. So to the left, top left is the behavior of this particular microtubule in GFP overexpressing cells. And so you will see that over time that microtubule moves around but doesn't break, stays the same, right? And now it doesn't grow and shrink because we are not looking at the tip, right? We are looking at the body of that microtubule. But then when we look at microtubules in the GFP ANC45 overexpressing cells, you will see that it bends and it eventually breaks. So the same pattern that we see in vitro, we can actually we can reproduce that. That's something we can see in cells. Okay, so now, and so we talked about microtubules, we talked about ANC45 and what it does to microtubules. Now, how about ANC45 in cancer? So ANC45 is overexpressed in cancer versus normal cells, and this is something that we knew from before. It's overexpressed in ovarian cancers versus normal cells, and it is overexpressed in other cancer types too. But what actually discovered is that ANC45 is particularly high in clinical specimens from patients that are resistant to taxol. So these patients, they have been treated with taxol and they have not responded. Interestingly, if we take a panel of ovarian cancer cell line that we know they have different IC50 to taxol, it seems that the cells that have the highest level of ANC45 are also the cells that have the highest IC50. We can also model paclitaxel chemoresistance in vitro because we can take cells and we can adapt them to live in presence of paclitaxel for several weeks. It takes several weeks depending on the cells you are looking for, but it certainly takes several weeks. And then when you get clones, either polyclonal or single clone that are resistant to taxol, and you measure the ANC45 expression level, you can see that the clones that are resistant to taxol also have a higher level of ANC45 as compared to the original population. So with this information in our hands, so we know that ANC45 is a microtubule severing protein. We know that ANC45 is overexpressed in paclitaxel chemoresistant cancer cells. So is it possible that ANC45 counteracts the microtubule stabilizing effect of taxol in cells? So to be able to answer to that question, we first had to look at what happens in reality in cancer cells when they are treated with taxol, right? So taxol is a microtubule stabilizing agent that needs to be given in vitro at nanomolar concentration doses, so to be able to mimic the clinically relevant concentration of the drugs. And so in absence of taxol, you have a metaphase cell, the cell is dividing, there is a fixed distance between the spindle pole, there is a ideal metaphase placed with, because that's the area in which the chromosomes are, and that's the area in which they have to be to segregate properly. And then as the microtubule, they start to pull apart, you have your chromosomes that are going to be aligned and they are going to separate in daughter cell, and you end up with two bipolar cells, each of which will give rise to the daughter cell. Now, when you are in presence of paclitaxel don't kill the cells as they are dividing the first time around, right? So, but paclitaxel causes hyperstabilization of microtubule. That leads to a number of phenotypes, including an increased interpolar distance, an increased metaphase plate width that results to misaligned chromosomes that are now pulled in many different directions because this cell is actually dividing in multipolar spindles. So your result really is of paclitaxel treatment is that as the cells go through cell division, some of them will keep living because they have enough chromosomes with enough genetic material to allow them to live, and some of them will die because they don't have the genes that are required for them to keep surviving. And that's why, one of the reasons why, unlike other drugs in vitro, paclitaxel really takes weeks and weeks and weeks to kill the cells if you're using the right concentration, which are low. So now the idea is, if ANC45 counteracts the paclitaxel stabilizing effect of the drug on cancer cells, then loss of ANC45 should phenocopy paclitaxel treatment, right? And that is precisely what we see. So these are cells that are dividing. We have done a knockdown of ANC45, and you can see that in condition of loss of ANC45, you have an increase in the pole-to-pole distance, you have an increase in the metaphase plate width, you have lagging chromosomes, and you have cell division on multipolar spindle, which eventually will lead to some cells surviving and to some cell dying. And so what does that mean in the context of ANC45 overexpression in cancer cells? Well, we think it means that when you have a tumor that have relatively low level of ANC45, you have taxol. Taxol causes increase of microtubule stability. The cells do what I show you the cells do in presence of taxols. They divide in multipolar spindle, and that will cause a number of cells over multiple cell division to die. However, if you have a high level of ANC45, taxol is not capable of stabilizing these microtubules, and so cells will keep dividing on will be bipolar spindled, and so they will just keep dividing, and that's how taxol resistance comes about in relation to ANC45. And so if we were to target ANC45 and treat with taxol, then we should restore the effect of taxol in absence of ANC45. And that is precisely what we do, or what we see. So we have cancer cells, these are spheroids, cancer cells, you treat them with taxol, you get some toxicity. These spheroids, they become smaller. Bear in mind that for us to be able to do this experiment, we can't keep taxol on for weeks and weeks and weeks, because when we're working with spheroids, you have a limited amount of time. You can keep the drug form if you actually want to evaluate these spheroids, and I think this is about 5 days, or either a week or 5 days. But certainly, if you are in absence of ANC45, so this is a knockdown of ANC45, already you have smaller spheroids, but then when you treat them with taxol, you certainly have a much better killing in absence of ANC45 as compared to the presence of ANC45. So the first conclusion from this would be that ANC45 can be a biomarker and the molecular target for paclitaxel-resistant cancer, meaning if you have a cancer with a lot of ANG45, you can expect this cancer to be less sensitive to taxol, similarly to what happens to tau, and you can also expect that perhaps targeting ANG45, we really don't know exactly how, but targeting ANG45 could restore sensitivity to taxol. So let's talk about now ANG45 in neurodegenerative diseases for a second. And so the work that you're going to see today was done by Yuri and Valentino, making lasagna, yeah? So the question is, does ANG45 play a possible role in Alzheimer's disease? And the reason for that is because I already told you that some of these microtubule-associated protein can really be playing a role both in cancer and in neurodegenerative diseases. And because by now, so by the time we attempted these results or these experiments, we knew that ANG45 is a microtubule severing protein, and we know that loss of microtubule mass is a characteristic of neurodegenerative diseases. So in this cartoon, what you're looking at the representation of a healthy brain to your left. So that's a healthy brain, a plump brain, and a healthy brain is a brain in which microtubules are coated by tau that stabilizes those microtubules, allowing them to have the structure that they need to have to function. And to the right, you have the representation of an Alzheimer's disease brain in which, among other things, tau came off, got hyperphosphorylated, came off of the microtubules to form a tangle of tau, which is one of the hallmark of Alzheimer's disease, and we will see it later in a picture. And most importantly now, these microtubules don't have the protection of tau anymore, and they fall apart. So to be able to answer the question on whether ANG45 plays a role in Alzheimer's disease, the first thing we needed to know is ANG45 expressed in brain? Bear in mind, when we started these experiments, we had no idea whatsoever whether or not ANG45 was expressed in brain. We knew by RNA data, published data, that ANG45 is expressed in all cells, but from RNA data saying that that is the case to whether or not it is expressed at the protein level, and if that is the case for brain, that's a long shot. So what we did is we looked at, those are mouse, this is a mouse, adult mouse brain, and it's stained for ANG45 for new N that is going to, maybe, let me do something different. Okay, so you are seeing it here. So this is the hippocampus. The area we are focusing on is the hippocampus. We are focusing in hippocampus because it has beautiful dendrites. So hippocampus has pyramidal neurons. So NeuN is this staining purple that stains for neurons because the brain has other cells other than neurons, right? So we want to make sure that we are focusing on neurons. And what you're looking at is dendrites that they come down here. And you can see they are really enriched in ANG45, which is this is endogenous, right? So it's a green secondary antibody. So that's a good thing. It's a good sign because ANG45 is expressed in the brain. And most importantly, it seems to be really, really bright and really, really accumulate or be present in dendrites, which are area of neurons that are, of course, rich in microtubule. Now, the other area of the brain that would be rich in microtubule would be the axons, right? So to be able to look at axons of ANG45 in axon, we did staining of the spinal cord. This is, again, mouse. So the spinal cord of a mouse, if you cut it horizontally, it's full of axons. So all that you see is axon surrounded by white myelin. Guine, I'm not sure how you could say that in English, but how do you say? Coated by myelin. There you go, that one. And so what you can see is this white myelin thing, and then the brown, this is immunohistochemistry, those are brown, those are groups of neurons cut horizontally. So you can really, really tell that ANG45 is largely abundantly expressed in dendrites and neuron, which is, again, a very good sign for us that ANC45 may be playing a role in regulating microtubule stability in the brain, and possibly play some role in the Alzheimer's disease. Next, we asked the question whether in cells, ANC45 colocalizes with microtubules. So those are primary cortical neurons that we get out of mice. This is endogenous, so we are staining with alpha tubulin to the left, top left, ANC45 top right, and then we colocalize them. We superimpose the pictures, and essentially you can see here very similarly to what we have seen in interface cells, in vitro cells. ANC45 really decorates those microtubules in primary hippocampal. We have done both hippocampal and cortical neurons, and we do see the same. There would be reason to see any difference in that sense. Oops. Okay, so this is just a magnification of the same picture. So this is in the cells, in the neurons, and this was in the purified microtubules. So very similar. So now the question would be, does ANC45 messes around with the microtubule mass in neurons, right? Neurons are not, are cells that are overall different from any other cell. So it's just best to make sure that that is the case. So those are primary hippocampal neurons. They are infected with either GFP or GFP ANC45, and similarly to what we have seen in cancer cells or in fibroblasts, the overexpression of ANC45 over a period of 48 hours, 24 hours leads to a significant decrease in the microtubule mass, suggesting that in neurons too, ANC45 overexpression causes loss of microtubule mass via severing. So now the question is ANC45 overexpressed in Alzheimer, right? The idea that we were testing was, okay, ANC45 is a microtubule severing protein. It is present in the brain. It is present in the brain in the area in which it will make sense for it to play a role in microtubule dynamic. Loss of microtubule mass is a characteristic of Alzheimer's disease. What about the expression level of ANC45 in these diseases? So what you're looking at the top, those are two Western blots. We just had a lot of samples, so we separated in two different Western blots. And so you have controls. You have MCI, mild cognitive impairment. So those are brains from patients that have mild cognitive impairment, so they don't have full symptoms of Alzheimer's disease, right? But their brain has the hallmark of Alzheimer's already. If you look at by via histochemistry, for example, and then you have Alzheimer's disease brain. And so when you actually measure the expression level of ANC45, we can see that ANC45 is overexpressed in AD, and it is already overexpressed in MCI as compared to the control. And when we look at where ANC45 is in those brains, you can see that ANC45 is in the amyloid plaques and is in the tau tangles. And those are areas where the tau came off, abandoning the microtubules, causing those microtubules to lose their mass. And so the localization of ANC45 in these areas suggests that, again, ANC45 may be actively participating in what happens to the microtubules in those areas. So that was positive. Even more positive was the fact that when we looked at other microtubule severing protein in the same tissues, we didn't see overexpression. So when we talk about microtubule severing protein, we are talking about 4 proteins now. We are talking about catenin, spastin, fidgetin, and ANC45, right? So when we looked at, for example, expression level of catenin and spastin, we did not see them to be overexpressed in Alzheimer's disease. So it really seems that ANC45 is a microtubule severing protein that is uniquely expressed in Alzheimer. Now, what makes ANC45 so different from the others microtubule severing protein? I mean, some of the experiments that we have done are very similar. The results of some of the experiments that we have done are very similar to the results you will get with the other microtubule severing protein. For example, the turf microscopy, the microtubule severing assay. So we see how it behaves. We still don't know how it does it. The fact, though, is that unlike all other microtubule severing protein, ANC45 does not have an ATP-ase domain and severs microtubule in absence of ATP, right? So ATP is present in the cells, obviously, but when we do turf microscopy, so when we take these brains that these microtubules that we derive from the brains and we put them in a chamber and we put ANC45 and we look at what they do, all other microtubule severing proteins do require ATP to sever those microtubules. ANC45 doesn't. And when we add ATP, nothing happens. They sever, ANC45 sever microtubules with the same frequency, nothing is changed. And so that is quite interesting because while the other microtubule severing use the ATP hydrolysis to pool, so if you see in this picture, so those are microtubules, these are alpha beta tubulins. The C-terminal of these tubulins, they stick out of the microtubules, and what the other microtubule severing protein do, they form hexamers, they sit on the microtubule, and they use ATP to pull the tail of those tubulins. And what they do, by doing that, they pluck out pieces of alpha and beta tubulin from the microtubule, and that causes the microtubule to fall apart because now it's missing pieces in the middle. ANC45 cannot possibly do that because it does not have this ATP domain. So the question is, so let me say something else first. So the fact that ANC45 does not have an ATP domain, ATP-ase domain, and works in absence of ATP can actually give this protein an advantage as compared to the other microtubule severing protein. The reason for that, because tumors, and particularly solid tumors, as well as neurodegenerative diseases, are characterized by hypoxia and low ATP levels. And this is a typo, characterized by reduced by hypoxia. Forget about the typo. But they are characterized by hypoxia and low ATP levels. So it is possible that a protein like 45 could work under conditions in which ATP requiring protein wouldn't. And so that could give ANC45 this functional advantage of other proteins. But that still doesn't explain how ANC45 can possibly break microtubule lattice and bend the microtubule lattice really without a force exerted by ATP hydrolysis. So the way we think it does is by binding to microtubule defects. Now, microtubules have defects along their lattice, right? That's normal to have them. But there are some microtubules under certain conditions that have more defects than others, right? And that is something that this is something that has been discovered really recently. Now, we always knew that microtubules have defect in their curved regions, right? So if you think about something that is quite rigid, and you bend it, of course there is a defect in that region. And these bent microtubules are often found either in very crowded area of the cells. They're crowded because either there are a lot of microtubules, or there are microtubules and other things like organelles, like mitochondria. And so literally microtubule would bend to get out of the way in this scenario. But also bent microtubules are very common in aging neurons, right? So fresh neurons, or young, I should say, young neurons have microtubules. They tend to be more straight. Aging neuron, especially in certain area, they can somewhat lose polarity at times and they become little bent. And so those microtubules have in their bent region defects. We also know, and this is a very recent work done by Melissa Garner here at the University of Minnesota, that microtubules exposed to oxidative stress, for example, hydrogen peroxide, will have defects. So oxygen peroxide will poke holes into the microtubule body. So does taxol. So because ANC45 is really present in these aging neurons, because it's a microtubule severing protein, it does not work via ATP, we ask the question of, is ANC45 preferentially binding to microtubules that we know have defects? And this is all very preliminary. So one of the experiments that Asumi, the pharmacology grad student, is looking at whether there is a different signal of ANC45 in microtubules that are bent versus microtubules that are straight. And in particular, she's looking at different ANC45 signal in one microtubule in a straight versus bent area. So this is red are the microtubules, green is ANC45, yellow is, of course, the merged image. And so you can see that in areas of microtubules that are bent, you have more ANC45 in comparison to areas of the microtubule that are straight, suggesting that ANC45 may preferentially bind curved region of microtubules for reasons that may have to do with the defects that are present in these areas. The same is true if we treat microtubules with oxygen peroxide. So at the top, you see again, microtubules in red and the yellow, which is essentially ANC45 on the top of them, in absence or in presence of oxygen peroxide. And you can see that the treatment with oxygen peroxide dramatically increases the binding, the signal, I shouldn't say the binding, but the signal of ANC45 on those microtubules, again, suggesting that ANC45 has a preference for microtubule defects that will be present following hydrogen peroxide treatment. And the same is true if we add taxol. So at the top 0, you have microtubules that have not been exposed to taxol. These are tricky experiments because sometimes we have to use taxol to stabilize the microtubules in vitro, but these microtubules are little artificial, so that we don't need taxol to stabilize them, for them to be stable throughout the 30 minutes where we do the imaging. But then we add either 10 or 100 nanomolar of taxol, and we see that the signal of ANC45 dramatically increases in those microtubules, suggesting that, again, ANC45 may be binding to defective microtubules that are typical of either curved microtubule, hydrogen peroxide treated microtubule, or taxol microtubules. And that is maybe the initiating factor that ANC45 uses to break, to sever microtubules in absence of ATP. So the conclusion from this portion, that again is very preliminary, is that ANC45 may be selectively targeting and acting on defective microtubules that are found either in aging neurons or in cancer cells for reason of hypoxia, hydrogen peroxide treatment, or paclitaxel treatment. Very briefly, I just have, this is my last slide. Therapeutic opportunities. We don't know how to target ANC45, right? But we know that ANC45 may form oligomers. We know that because other microtubule severing protein do that. Catenin does that. Catenin forms hexamers, so 6 catenin, they get together and they kind of sit flat on the microtubules, and they have to have that six of them, and they have to sit on microtubules in a particular way to be able to pull these tails the tubulin tails to be able to make the microtubules fall apart, and that's what you're seeing. You're seeing at catanin here. So it says 6, it sits and it pulls. So it is possible that ANC45 may act similarly. In fact, the warm isoform of ANC45 forms dodecomers. That's what you're looking at here. So we preliminarily looked at whether or not recombinant ANC45 forms oligomers. And so we're looking at a monomer of ANC45, and then we add a different concentration of cross-linkers. And you can see that you have the formation of high molecular species ANC45 in presence of the linker, which is something that you have to use in vitro anyway. So otherwise, the interaction is never going to be strong enough for you to be able to see. But that suggests that ANC45 also needs to oligomerize to some extent to be able to perhaps bind to microtubule and to function and to break them. So the idea is that if we can understand which areas of this protein are required for oligomerization, and we can prevent either oligomerization to start with, or can prevent the binding of the oligomers on microtubules, depending on whether ANC45 oligomerizes before or after binding to microtubules, then we could potentially, theoretically, be able to target ANC45. And that was my last slide. So I'll be happy to take your questions if you have any. Thank you.
Great. If you have a question, please raise your hand and I'll bring the microphone.
It's easy to see, it's not too crowded.
Thank you, Martina. That was a great talk.
Thank you.
I'm curious about what's driving ANC45A expression, like in your ovarian cancer cells.
I can hear you, yeah.
Okay, in your ovarian cancer cells that, the chemo-resistant ones have more ANC45A. Do you know what's driving its expression?
No, I don't. I don't know if it is something that happens as a response to the capacity or incapacity of cancer cells to do something, but we have no clue, no. Also, we don't know, it's a little bit more complicated than that because we looked at the expression, right, which is the easiest thing to look at. But we don't know about activation either, because now a protein that in vitro or even when we're overexpressing has such a strong capacity of severing microtubules, like you overexpress it in cells, and within 48 hours you have no microtubules left, that is not compatible with life, right? So obviously these ANC45 must be regulated, right? The activity of this protein must be regulated, and most of these proteins are regulated by phosphorylation. So there gotta be a way in which this protein is on and off, depending on what the cells need to do at any given time.
Yeah, I'm wondering about like during the cell cycle too. Maybe there's some cell cycle mediated regulation.
Very likely, very likely. We have just chosen to, for the moment, we have just chosen to take the direction of looking at the more cell biology of the protein per se, because we know, I feel we know too little about it to be able to make reasonable assumptions of what it may do through the cell cycle in cancer. It's a little bit of a slippery slope according to NIH reviewers anyways, as you can suspect, which is fair. I think is fair. We know too little to be able to guess in a way that would make sense. Yes, yes.
Hi. Yeah, that was a great talk and beautiful images. I guess similar to Kaylee's question, you had mentioned that as neurons age, they have more bent microtubules, and I was wondering if there was anything in the literature that would suggest also an increase in ANC45A as people age. I assume your patient data, they were all aged match, but like that would have been a good place to look at.
Yeah. No, there is no data in literature because there is nobody has ever shown ANC45 to be expressed in brains to start with. So this data, the ANC45 in neuron part that you see is unpublished. And so nobody has ever looked at whether the expression level of ANC45 in the brain changes over time. It doesn't seem to be the case based on our, 5 and 7 specimen, or 5 plus 3 plus 7. But yeah, I don't know that. Daggy, yeah.
That was great. So no one has a more detailed structure of like crystal or cryo-EM of ONC45A sitting on.
No, we are trying to do it.
Okay.
Yes. So the idea now would be to, and we are trying to start a collaboration with Ilaria Testa. She's a microscopy expert at the Royal Institute of Technology in Sweden. And what they do is basically, they do a kind of microscopy, it's called stealth microscopy, maybe wrong, but basically what they do is they have the resolution of cryo-EM, but they can do it in live cells. So it's a combination of turf and some other things that allows you to Armstrong resolution. So you can actually see what happens to a protein in a cell as compared in correlation to the whatever structure that you're looking for, in our case, microtubules, without having to fix the sample, which is what you're going to do. You are going to have to do in electron microscopy. But that would be the next thing. The idea is to take microtubules to damage microtubules. So when you damage microtubules, you can literally see the damage, and then see where ONC45 goes as compared to the damage. Does it go on the top of the damage? We think it goes next to the damage, because once a microtubule damage is made, given the opportunity, meaning if free tubulin is present in the system, that the damage will be repaired, right? So the question is, why would ONC45? Because if ONC45 sits at the interface between the damaged and the undamaged, there is where there is a protofilament shift. So that is an inherent damage of a microtubule that has been repaired. So to answer to your question, no, nobody has done it. We are going to try to do it.
So a second question, have you looked at RNA or protein expression in primary cancer specimens to see if there's any correlation with taxol sensitivity, the patient, and because I know that you guys or Tim had created a lot of avatar-like things in PDX models.
That is on the laundry list of Tim, yes. So it's one of those things where the confounding factor is that most of patients have gotten both. Taxol,
yeah, and carbo.
yeah. Yeah.
Does RNA correlate with protein?
It does.
Okay, I'll send you an email, because we have a lot of breast cancers that were treated with taxol only, and
Yes.
we know something about the sensitivity.
Yes. I think this is something we talked about years ago. I don't know, and then we got lost. But yes, the idea was that because breast cancer patients are treated with one at one point in time, anyways, that the correlation is more straight. I mean, for us, when we looked at what happened to the cancer cells that we were adapting to taxol, at one point in time, we even suspected that cells would become resistant to taxol because of the pump. But we looked at, we did HPLC, and we looked at the amount of taxol inside the cells is the same. So it's really not, and there is no difference in sensitivity to cisplatin. So that's why we could argue, I guess, that whatever we saw in that system had to do, that was related to ONC45, had to do with taxol and not with cisplatin. But yes, the breast cancer patient will be a more straightforward correlation, clinical correlation, yes.
All right, any more questions? Then let's thank Dr. Bazzaro one more time.
Thank you so much.